Okay, thank you. So I'm, as I was introduced, here to talk about the universal genetic code. And the way I normally introduce this project to pure biochemists is I first have to convince them that this is something that might have evolved and did not magically appear overnight, but I think I don't need to convince this room of that. The very earliest life likely used a genetic code that was more simple than the universal code we know today, likely comprising of just a handful of amino acids, and it complexified over time, adding more and more amino acids as it went. There are a bunch of different theories for the order in which these amino acids were added. These theories include the number of codons they have, the GC richness of their codons, the chemical simplicity of the amino acids themselves, and whether they've been discovered on meteorites or synthesized in the lab. Finally, we can look for clues in metabolism, so the order in which they can be synthesized in biology. So there's an excess of 60 of these different hypotheses for the order of the code's evolution, and for my work today, we're utilizing a meta-analysis performed by Trivanov in 2004, and he took all of these 16 amino acids and synthesized them into a consensus order, and so this here represents a single hypothesis for the code's evolution. And today I'm going to be interrogating that and at the same time interrogating a more broad hypothesis. Can early genetic codes produce proteins that can support functions that are essential for life? And to do that, I took theoretical snapshots through this hypothesized consensus order and generated libraries of randomized proteins that represent these different hypothetical codes throughout the evolution. So my most simple code has only the 5 most ancient amino acids, and then the 9, and then the 16 lacks just those final 4 amino acids, and then we have a positive control code, which is all of today's amino acids. So the protein variants in each library have a randomized region of 80 amino acids, and each library has a complexity of greater than 10 to the 12 unique sequences. So the power of our approach is that we planned to compare these 4 libraries, these 4 alphabets of this evolving code, and see what propensity they have for structure and function. So the first thing we did was interrogate for structure, or a proxy for structure, which is the ability to form soluble protein when expressed in E. coli. So I took about 24 individual variants from each library and expressed them, and then used simple soluble versus insoluble as an idea of whether they can fold in the cell. And a rough conclusion is that the newer alphabets, the 16 and the 20, were most likely to be expressed, whereas the older alphabets, the 5 and the 9, when they were expressed, were more likely to be soluble, which was interesting. So now we had our sometimes soluble proteins. The next thing we wanted to interrogate was which of these alphabets, or do all of these alphabets, have a propensity for functions that are essential for life? So one of the most simple functions I can select for, because I kind of wanted to go a little bit easy on these alphabets, is ligand binding. And some essential ligands at the origin of life, as Mark has already mentioned, ATP and GTP. So these are essential energy currencies for all of life, and also components of RNA, which I think we can agree is somewhat central to origin of life. So the method that I use to interrogate these libraries for binding of these cofactors is an in vitro evolution method, again similar to Mark, called mRNA display. For those of you who aren't familiar with it, it's a technique that in which you physically attach a protein variant to its own encoding mRNA. And this is really powerful because it means that you can take a library of trillions of unique sequences and you can pluck out a single protein that has your desired function, and because it's attached to its own code, you can decode it and get its sequence, and you can also propagate it through PCR. So aside from that, it's pretty similar to the technique that Mark spoke about. So we generate our RNA protein fusions. We introduce them to some immobilized ATP and GTP ligands, and then only the fusions that can be competitively eluted go on to form the precursor for the next cycle. And you assume that once you have binding, you observe increased enrichment after every round. So I first performed this with our control alphabet of the extant 20 amino acids, and you can see along the bottom here the number of rounds and then the percent of fusions that are selected per round up on the y-axis. And this was really encouraging to see because after every round we saw increased enrichment for ATP and GTP binding. You can see that it is significantly above a negative control that I did. But this was my positive control, and I didn't know how the other alphabets were going to behave. 5 amino acids is awfully few to expect much out of. So I repeated this experiment with identical conditions for the 3 other ancient alphabets, and was really surprised to see that all 3 of the reduced amino acid alphabets yielded ATP and GTP binders after an average of 5 rounds. So this was, it may not have enriched quite as high for the 5 library, but that's definite enrichment, and this was a repeatable result. So really excited about this. So the next stage was to find out what these populations of sequences are. So I picked a round from each of the experiments for deep sequencing. To glean a little more information from this experiment, I repeated all of these rounds, and then after I generated the fusions, I split them into 4 different conditions. So the first has ATP and GTP pulled together, as my typical selection round was, and then I did ATP by itself, GTP by itself, and then a no ligand control as my negative control. I had the kind help of Celia Blanco and Irene Chen, who I think are here, in analysing this data. And the metric we came up as a proxy for binding affinity was relative enrichment. So we considered an individual sequence to be enriched if it had more copies after selection than it had prior to selection. And we considered it to show relative enrichment if this enrichment was greater than this enrichment for the negative control. So we would expect the values for relative enrichment to be greater than 1 to show real specific binding. So this is an awful lot of information, but I can walk you through it. Each of these circles represents a unique sequence. On the y-axis, we have relative enrichment on a log scale. Along the bottom, we have the 3 different selection conditions per library, color-coded up here. The line here is the threshold for relative enrichment. So everything above this is showing specific binding. Everything below we don't want to think about. The median value is shown in black. And straight away, if we look at it, we can see this trend of increased propensity for binding and increased affinity of binding with the increased complexity of the alphabet. But there's a plateau after you reach the 16 amino acid alphabet, which suggests that those final 4 amino acids aren't adding a lot for this particular function and this particular selection. If we look at the 5 library, those medians are really low. There's still some binding, and the best binders, yeah, they're showing some relative enrichment, but if we look at the 16 for ATP, it is several orders of magnitude better than the 5. And that was really exciting to see. I'm going to zoom in now on the best 6 binders from each library, from each alphabet. And again, this really pronounces the difference in relative enrichment between the alphabets. So you can see that you have a relative enrichment between 1 and 10 for the 5, and all the way up to over 1,000 for the 16. But this is for ATP, and this pattern isn't as evident for GTP binding. So the final thing I wanted to do, talk about the specificity of binding. As I mentioned, I performed the selection with a mixture of ligands, mostly because I couldn't be bothered doing it twice. But it's quite striking if you look again at just these top 6 selected binders per alphabet. You can see that for the 5 and the 9, there isn't really much discrimination between binding. So the ratio of ATP and GTP is on the y-axis here. So there's some really sloppy, non-specific binding happening. Whereas if you look at the 16 or the 20, there's much more of a bias one way or the other towards ATP or GTP for the 20. And this suggests that as the chemistries of the genetic code get more complex, they are more able to distinguish between the ligands that they're binding, which is, of course, a function that is crucial for modern biology, or any biology. So I would like to go to some really quick take-home points. We managed to find binders from the 5 amino acid alphabet, and that is something we never expect to see, and we're really excited about that. We observed that the relative enrichment, the proxy for binding, increased with library complexity. The best binders were from the 16 library, not from the 20, and the most selective binders were from the 16 and the 20. So I think I can tentatively and maybe cheekily conclude that under some very specific conditions, hypothetical early genetic codes may actually rival today's universal code. With that, I'd like to acknowledge people who've worked on this and ask for questions. Thank you.
So we have time for a few questions. Yes, please.
Yeah, so I'm very interested. It looks like your 9 amino acid data set was not quite as well behaved as some of the others. And so it looks like it took you more rounds of amplification to get, a high yield. And then also, when you were showing the binding capacity, it looked like there was more capacity for not such, stringent binding. And so I'm just wondering, to me, like interpreting that, I don't know, maybe there were some logistical problems there, but interpreting it in more of an evolutionary context, it looked like, that 9 amino acid alphabet may not have been, a particular advancement for early life, that there was a bottleneck there or something. I wonder your interpretation of this data.
That you've identified a stone in my shoe. Yes, the 9 did take longer, and I repeated it, and it still would consistently take a little longer. I don't have a good answer, because when we did get binders, they are identifiably better than the 5. One technical caveat is that the complexity of the 9 library at the beginning of the experiment was higher than the other 3 libraries. So that is something that we need to correct for when we publish this, and it might be that actually explains for that extra lag before we saw enrichment. It had more sequences to pair through before it went right up.
We have time for one more short question. Mike?
So did you interrogate the mechanism of binding? Because I thought when the Shostak company did the first of these types of experiments, when they finally did some crystal analysis, it turned out that the binding mode was backbone interactions, not through side chains.
We're in the process of doing that right now. Okay.
Okay, let's thank Matilda one more time.